So I think we are ready for this chapter, Introduction to Immune System. And today I will just discuss about few things, which are definitions. The chapter starts with a few definitions, and if you really start like that, it is going to be difficult. But we will follow Harrison. Okay, so that makes it quite easy for all of us. So if you really see, they have given definition which are in alphabetical in order. And that's quite odd, right? Because this is first chapter, and if you really don't know about immune system, it becomes difficult to go alphabetically. It should have some sequence that okay, it starts with innate immunity, then adaptive immunity. What two terms I told are the main two arms of the immunity: innate immunity and adaptive immunity. We have learned this in second year MBBS, but not that sincerely, right? The microbiological part got more importance, and the immunologically somehow we did not learn into that detail, specifically being scared of those CD numbers and cytokine numbers and what they do and how they do, right? So that becomes the challenging part. And today we want to address this chapter. This chapter is quite extensive. So we'll be going parts by parts. So today is the definition day. So we will just see. It's not the definition what Chatur says in Three Idiots. It's a simple definition what we are trying to learn, and I'll just introduce the concept where I think some clinical application I can explain it to you. So the very first definition, if you see, the definition goes for the adaptive immune system. Now the two keywords you will remember, adaptive immune system, are cellular immunity and humoral immunity. Cellular immunity is by T cell, humoral immunity is by B cell. Rest of the things we will take care. It's a specific system, more robust, developed later in the evolution, and it has that memory which innate does not have much. It has that specificity which innate does not have much. So that's the difference of adaptive immunity and innate immunity in a very broad sense. We will be going in the chapter as the chapter evolves. So that was the first definition. We should have an idea about that. Then they talk about antibody. Now, see, antibody is very important. That Y-shaped structure, what we all have seen and what we all have witnessed the part. But here also, I will say the antibodies are like machine guns in the immune system. The B cells are the major producer of this antibodies. It requires a lot of processing and all part, requires a lot of rearrangement, and then you get that Y shape with the heavy chain, light chain, and the total structures are formed. And you, this is IgG, IgA, IgM, IgD, Ig. There are different types of antibody. And these antibodies can be there present on the cells like B cells. They can be in circulations. They can be at the mucosal level. They can be in the body fluids, like it can be in saliva, it can be in CSF. So it can be at many places, and that we should be knowing. So this was about the antibody. Then they talk about something called antigen. Now, this antigen can be either foreign molecule coming from the microbes or self molecule itself, which are altered. Okay. Now, something to be called antigen, it is not enough to be foreign or something altered self. It has to trigger immune response. Okay. So then only you call it antigen. Okay. So that is very important to remember that immune cell triggering should happen. Then comes antigen presenting cell. See, now what happens is whatever the damaged part of the antigen, what we told that it can be either the foreign part or it can be the part from the altered self, right? Altered self will lead to autoimmunity. That is the part we are broadly trying to remember part. Okay. So this antigen needs to be presented after the initial identification to a more robust system. What is that more robust system? Adaptive immunity. Okay. Now for the presentation, you require certain cell to do this job, and that is called antigen presenting cell. If someone asks what is the key antigen presenting cell, answer it dendritic cell, but it is not function limited to dendritic cell alone. The antigen presentation can also take place with few other cells also, specifically macrophage and B cell. Okay, let's form an MCQ. All of the following are antigen presenting cell except, right? Then three right answer will be dendritic cell, macrophage, and B cell. So these are the three antigen presenting cell. Why dendritic cell works? In MCQ, dendritic cell, macrophage, and B cell. In body also, this three cell works. Okay, so this is how we should be remembering that part. Then there is a definition of apoptosis. We know programmed cell death, right? That fast receptor will come. CD95 will be the term which will use. TNF receptor will come, and the fast ligand, and then the pathway cascade start with the caspase family and all, and the cell dies. Here, cell dies without making much noise. The noise is made in necrosis. In apoptosis, it's a programmed cell death. So when it dies, the body does not react to that. While in necrosis, when the cell dies, body reacts to that. Now, very important concept I want to put it here. When the cell dies, it is not enough that the death has to be cleared. If it is not cleared for the longer time, the dead cell stays there exposed to human immune system. It can make antibody against the structure of the dead cell. They are our own structures. They are our own structures. So autoimmunity. One mechanism will be there in autoimmunity that apoptotic debris are not cleared efficiently. That can lead to autoimmunity. When you will read lupus in detail, this will be one of the mechanism. The defect in apoptotic pathway can lead to autoimmunity. So the things are beautiful. Okay, we have to clear, we have to understand, and that's how the things are going to work, right? So now I will go to the next definition. That's autoimmune disease, right? So autoimmune disease is something we broadly tell when the adaptive system goes crazy and attacks our own cell, it is autoimmune disease. The next definition will come is a autoinflammatory disease. When the innate immune system goes crazy and attacks itself, it's called autoinflammatory disease. See, in your autoimmune disease, there will be antibodies because they are B cell, T cell mediated diseases. Okay, so there will be antibodies. Like example, lupus. Your autoinflammatory diseases are little different. Here, the problem happens in the innate immune cells. Like you have neutrophils and all, intrinsically they get activated through some trigger or through the genetic problem itself. Once these are activated, there will be a lot of production of interleukin 1, interleukin 18, interleukin 6 and all, TNF and all, and that will lead to fever, joint pain, skin rash, and so many manifestations. So clinically, it looks almost same, but there is a pattern to identify. There is an onset of age to identify. Absence of antibody, not truly, but yeah, broadly, absence of antibody in innate system. These are all autoinflammatory syndrome. What is the best way to learn that? In your Harrison, the chapter list in rheumatology, the last chapter is autoinflammatory syndrome. That tables is all autoinflammatory. The before that, whatever you are saying, most of them are autoimmune problem, lupus, rheumatoid arthritis, systemic sclerosis, mixed connective tissue disorder, Sjogren's syndrome. They are broadly the autoimmune diseases. And we all have been taught autoimmune more. Autoinflammatory is in the picture, and we are seeing a lot. One example of the autoinflammatory, I won't say autoinflammatory, but triggered inflammatory, which use the innate mechanism, can be gout. Gout has MSU crystal. This MSU crystal can trigger the inflammasome activation, and then the pathway starts for activation of IL-1 and all. So canakinumab, rilonacept, and Anakinra, which targets IL-1, are approved for gout, right? So that is your autoinflammatory disease is what we are talking about, or the innate system disease is what we are talking about. While you're on adaptive, you're talking about the lupus and all part. Great. Then comes autophagy. Autophagy, you have to remember, it was more of a clearance mechanism that inside the cell, if things are not working that great, there are debris, there are damaged cell, we just want to eat that out and we want to clear it off. Okay. But if that clearing process makes a problem, then again, the cell can be at stress and autoimmunity can be triggered. Autophagy has one more important point, the intracellular bacteria, like mycobacteria, that can be taken care by autophagy. So autophagy defect can lead to susceptibility to all this infection also. So that's autophagy. Okay. Then comes B cell receptor for antigen. Now BCR is nothing but on B cell surface, what receptors are there? It's called B cell receptor. For antigens, specifically those receptors are nothing but the antibodies. This express this IgM on that, IgG on that, IgD on that. Okay, so these are the BCR cell surface receptor or the cell surface receptor for antigen. Okay, then there is a definition of B lymphocytes, like the cell which comes from the bone marrow and through the hematopoietic stem cell. That's a B lymphocyte. T lymphocyte also comes from bone marrow. They get trained in thymus. That's why they are called T lymphocyte. Then there is something called B regulatory cells, right? In body, there is always the mechanism of homeostasis that B cell makes antibody, but there needs some control over that. So B regulatory cell controls that part. Okay. Whenever you are learning the control, remember one cytokine, IL-10. There are a lot, but remember IL-10 for sure. IL-10 is more of a regulatory cytokine. And some are fighter cytokines, like your TNF and IL-6, IL-1. They're fighter cytokine. There are certain cytokine which can play two roles. IL-2 in a higher level and lower level has different roles. Okay, so things are crazy in immunology, but overall, don't try to remember. Just try to see how things are working. You will remember. I am not telling that because in MCQ you can't tell that I enjoy concept. You have to answer, right? So that's what we will be doing with that. Then comes CD classification. See, now in CD classification, I want to introduce one MCQ, that what was the landmark Nobel winning discovery which actually led to this CD classification. It was about monoclonal antibodies. Okay. Then we realized that we can have those monoclonal antibody to each cell, which can be labeled by the CD markers against that protein, this protein. So all these clusters of differentiation CD present on the cell can be identified by those monoclonal antibody, and then it can be targeted also, like rituximab targeting CD20 on the B cell, right? That is your CD system. Okay, a lot of CD system and CD numbers, and that was also one reason we did not read immunology in detail in second year MBBS. If we don't want to go for numbers, right? So that was fine. Now comes few cells, CD4, CD8 cell. So CD4, I always tell, is like a boss. CD4 orders that B cell produce antibody, CD8 go and kill. CD4 is more of a boss. Okay, then there are subtypes of the CD4. There's a TH1, TH2, TH17, T regulatory, TH9, TH13. We will come to all this part. Okay, but CD4 as such is the boss. The job is to order or to secrete cytokines and make the surrounding system fight worthy. Okay, so that is the job of CD4. CD8 is a real soldier. It goes, attack the malignant cell or virally infected cell, eats it out, and sometimes it dies in the process. Okay, so it's a real soldier, right? CD4 is the boss. It orders. Okay. Then comes the part chemokines. Chemokines are nothing but they are messenger which communicates the total process. So chemokine has one part, movement and circulation of the immunity or the immune cell milieu, and then it works beautifully on that. Then comes the next part, complement. So complements are circulating enzyme, where they are circulating in plasma, where they are produced majority in the liver. Okay. So what it does, it makes the process faster. Okay, so that's a complement cascade in that. We will go into detail of that later, but right now we're just touching through the definition. Then comes the post-emulatory molecule. A very good MCQ part, post-emulatory molecule. Okay, so what happens is, see, the cell has to interact with each other. So when they're interacting with each other, like presenting antigen, then making a response or immunological synapse of innate and adaptive. So they both come from one side and second side, and then the building happens very properly, and then only the body reacts to those parts. So that is called something what we tell post-stimulatory molecule. So you know antigen presenting cell now. I know you remember that. I told one was main was dendritic cell. Second was what? B cell. Yeah, great. And what third was macrophage. Okay. So now this antigen presenting cell will have co-stimulatory molecules like B71, B72, or CD40. Okay, that is the present on that. What we told, B71, B72, or CD40. Why I'm talking like this? Because these are MCQs. We have to remember they are on the APCs. Now these APCs are what? Dendritic cell, B cell, or the macrophage. They are going to present antigen to adaptive system like T cells, right? So on that there will be certain APC because they have to then make a proper immunological synapse. So what will be their things? There is a CD40 here on the APC. So what should be there? CD40 ligand. That easy. Okay, CD40 ligand or CD28, right? So that is the co-stimulatory molecule on that. Now just try to remember there are drug targeting all these antibodies, CD40 ligand, CD40 and all, and they are the co-stimulatory blocker. You must have heard even CTLA4, which is one of the co-stimulatory or the immunological synapse molecule, right? So that also comes in the picture, and you remember the drug called abatacept, right? So that comes in that picture. Now there is a definition of crystallopathies, like which crystal we just talked or we know, MSU, monosodium urate. It is not uric acid crystal, it's a MSU crystal. Okay. Now there are many other crystals. There is a BCP crystal, CPPD crystals. Okay. Now oxalate crystal. So this crystal intracellularly, if they activate the inflammasome in the innate cells like neutrophil and all, it will lead to production of IL-1, IL-18, and then the inflammation will go up and the diseases can come, which can manifest like joint pain or the pain surrounding the joint, periarticular pain and all. They can be in other areas also, but these are called crystallopathy. Gout is a prime example of that. So if you remember gout, it is explained here. And then when we have a total chapter on the crystallopathy in the Harrison, so where we discuss this, that will come into picture. But these are the crystallopathies, right? Gout is the prototype example of that. Then comes the term cytokine. A difficult term, right? We all know cytokine is a difficult term. But again, what are they? They are communicator. They are basically proteins. They are communicator. There are so many numbers, IL-1, 18, and you can tell a lot of number there. What they do? They communicate information for the immune system. Okay. Because see, antigen has come or the microbe has come and we are going to attack and all this all are fine. But every cell which is getting activated ultimately will result in the gene transcription, which will lead to production of the cytokines. That will be telling message to other immune cells, and somehow this is the network which gets established, right? The message is getting conveyed by chemokines, and the work is really being done by cytokine. So, chemokine, cytokine are very important points for the immune system, how it is going to work and how it's going to react, right? I will go in detail of those things, okay? Or we might not go even, it's fine. Because see, when you actually read the subject in a proper detail, when you correlate it with the patient, then it starts making sense. If you are trying to make sense line by line in a textbook, I think we are in the wrong business. Okay, it is difficult. For MCQ, we won't miss. Don't worry. I'll cover the points which are required to be covered for that. Okay. Now comes the dendritic cell. So we talked about that, right? Dendritic cell are the major antigen presenting cell. It is the synapse between innate immunity and adaptive immunity, right? So that is a part you have to remember. Then there are types of dendritic cell. There's a myeloid dendritic cell, then there is a plasmacytoid dendritic cell. So that is the overall part which comes and it has then again roles. And when we go in the detail of innate immunity, we will come to this. But it is considered a part of innate immune system. The major job of this is a messenger to go for. Then there is something called Ig fragment of the Fc, okay, Fc receptor, which comes. Now see the Fc. We have remembered that Y-shaped structure of what we say antibody, right? The Y-shape. Okay, that was crazy. So we have remembered that Y-shape structure of the antibody, and in that Y, what the down comes, that arm of the Y, it's called Fc portion, right? And what Y comes, it is what? It comes as the Fab, that is the antigen binding portion, right? So you have the antigen binding portion, you have the that Y standing line portion, right? Now that portion of Fc actually gets attached to Fc receptor. Now, where are this Fc receptor located? On the immune cells, obviously, because then antibody binds to that, and then the further activation down signaling happen in the cytoplasm. So this is very important. These Fc receptors are located on the B cell, NK cell, macrophage, neutrophil, and mast cell. Someone might make MCQ out of that, sorry, but that is a possibility. So Fc receptor are located on which cells? So if you really talk about the adaptive immunity, they are located on the B cell. B cell. Adaptive immunity has which cells? B cell and T cell. So out of that, the Fc receptors are located on which cell? B cell. The rest are all your innate cells like NK cell, macrophage, neutrophil, and mast cell. One of the mechanisms, something called antibody dependent cell mediated cytotoxicity, ADCC. For that, these Fc receptors are responsible. Then this receptor are named also. You can see your Harrison part into that there is a CD16 which is called Fc gamma R3A. Then there is a CD23, it's called Fc epsilon R, it is for IgE, Fc epsilon R. Then there is CD32, which is Fc gamma R2. Then there is a CD64, which is Fc gamma R1. Then there is a CD89, which is Fc alpha receptor. Okay, so these are the receptor and the names important. Sometimes it is important MCQ. They have different function also. But whenever we are talking about Fc gamma, we are mostly talking about IgG. When we are talking about Fc epsilon, we are talking about IgE. So they are expected to be present on what? Mast cell, Fc epsilon wala part, right? Then there is a concept written inflammasome. Now, inflammasome is a cytoplasmic structure which as such does not exist in cytoplasm, but when the innate system gets activated, it gets arranged in the cytoplasm. It gets arranged in the cytoplasm. Okay, so that is an important point. It is a link sensing the microbial product and cellular stress to the proteolytic activation of IL-1 beta and IL-18 inflammatory cytokine. Okay, so whenever there is a problem inside the cell, it has to get alert, and the alert should result in inflammation. For that, whatever link is being prepared by the rearrangement of the structure inside the cell are the inflammasome. Okay, so that is the part inflammasome comes. Now they have given definition of innate system. You know innate system now. Adaptive is evolved system. Innate is there for a very long time with us. So that's the innate system. What it does, it has a very non-specific but very broad role. It identifies almost everything. It senses the danger and immediately it tries taking care of that, and also starts conveying information to adaptive that I will need help, so please help me. But initial part, I am taking care of that. Okay. So the cells which are taking care part of, you will always remember neutrophil will come in picture, right? So neutrophils are very important structure of the innate immune system. Of course, along with that, you have basophil, eosinophil. So, right. So we, whatever your pathology fields were there, neutrophil, eosinophil, basophils comes into that picture. Then you have tissue mast cell, you have epithelial cell, you have dendritic cell, you have macrophage monocyte, you have NK cell. This all are part of innate system. The very important structure they have is the pattern recognition receptor, PRR. One example will be TLR4. We all have learned in the septic shock, right? That lipopolysaccharide will be identified by TLR4, and the further cascade continues. So that is your TLR4 for the same reason, right? Then comes a very important concept, innate-like lymphocytes. See, when we talk about lymphocyte, our mind goes to lymphocyte means adaptive, right? B lymphocyte, T lymphocyte. But there are certain lymphocyte which are not adaptive. I already gave one example in the previous innate system, NK cell. They are lymphocyte. They are not adaptive. They are innate. So there are few other cells specifically at the mucosal level, gut level, and the bronchus level and all, which do function like adaptive, but they have main characteristic of the innate. Okay, so these are called ILCs, innate like or the innate lymphoid cells. Okay, so that is very important. Now there are types in that also. There is like ILC 1, 2, and 3. ILC1 is something like CD4TH1 cell. ILC2 is like CD4TH2 cell, where ILC3 is like something TH17 cell like that. Okay. Then comes the natural killer cell. Now, this natural killer cells are something called surveillance cell. What it will do? See, in body, majority of the cell expresses HLA class 1. Okay. So if the HLA class 1 is not expressed because of the some problem inside the cell like malignancy or the virus infection, the NK cell will eat that up. That's why they are called surveillance natural killer cell. Okay. Then there is a combination also in innate variant only, NKT cell. So they will have almost a combined function like that. But the most important MCQ will remember about NKT. What does it identify? It identifies lipid antigen of bacteria, fungus, protozoa, and the virus. What does it identify? Lipid antigen. And that is your MCQ for that. Then there is something called PAMP, pathogen associated molecular protein. Because see, there are so many bacteria, so many fungus, so many viruses. They have certain common structures. Those common structures can be identified by the innate cell. That's called PRR, pattern recognition receptor on the innate cell will identify common structure on the bacteria. Like gram positive has something, gram negative has something. Then there are some other structures. So these are called PAMP, pathogen associated molecular protein. For example, I can tell like again lipopolysaccharide, right? So that is the PAMP. Even the double-stranded DNA, single-stranded DNA, RNA, this all are PAMP. you will have a pair of PRR to identify that. Okay, so that becomes the structure there. Then you have T lymphocyte, obviously the adaptive cell, which are produced in the bone marrow, gets trained in the thymus, comes into circulation, and does the job for adaptive immunity, cellular immunity. That's a T lymphocyte. Then there is a status called T cell exhaustion. We have heard of this in which disease? HIV, HCV, and sometime in the malignancy also. What happens is the persistence of antigen. If something uh, antigenic is continuously there, the immune system will be constantly fighting to that. But in that fight, it will get tired. That's called T cell exhaustion. After that, it won't be able to make memory even to the other antigens and all. So our susceptibility to infection will increase. See, in HIV, you have seen that, right? So there is a hypergamma means antibodies are high in the circulation. You are more susceptible to infection. Patient is more susceptible to infection. So that is the odd part there. So that is something what we can T cell exhaustion for that. Okay. Then there is something called TCR for antigen, T cell receptor. As B cell has the receptor, T cell also has receptor. Now see, this is the most important point. The BCR on the B cell and the TCR on the T cell makes it adaptive system. If lymphocyte does not have structure like BCR or TCR, they become the innate mostly, like NK cell. Okay, so this is the lymphocytes thing. Lymphocytes are all same. The surface makes them either they are in adaptive by the presence of BCR or TCR, or in innate by the absence of these two, right? So that is how the things are working. Then there is something called TFH. Okay, T follicular help cell. They are in the germinal center, lymph nodes and all, spleen and all. Okay, what is their job? Their job is very important. They want to make antibody refined. How does they do that refination? By a proper B cell differentiation and affinity maturation. Okay, so we know that adaptive improves by the attack to attack, thanks to this T follicular helper cell. Okay, now this T follicular helper cell has particularly two cytokines, IL-4 and 21, which makes in helping all this affinity maturation and all part of antibody properly. And it is actually regulated by MCQ, which is called BCL6. Okay, the BCL6 is a transcription factor which regulates T follicular helper cell. It has a major role in IL-4 and IL-21, and then it does its job for affinity maturation and B cell differentiation. Affinity maturation, T follicular help cell in germinal center. Fine. Okay, fine. Now you have TH1, TH2, and T regulatory, TH9 and TH13 and TH17. These are a subset of the CD4 T cell mostly. Okay. Now we will go to that little in more detail. Now CD4, TH1. So TH1 is more of, for each cell you should be remembering what is the transcription factor of that, what are the cytokines of that, and what are the role of that. Three things, right? What is the transcription factor which regulates that? What are the cytokines this cell secretes? And what is their role in the immunity? If you remember three things, you will be thorough with that. Okay. So TH1. So TH1, the transcription factor, it is T-BET. 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 Okay. What are the main cytokines by this TH1 cell? It is interferon gamma, IL-2, TNF beta. Interferon gamma, interleukin 2, and TNF beta. It has a major role in cell-mediated immunity, right? CD4 is a cell-mediated immunity. Then there is a TH2 variant of that. Now TH2, what is the transcription factor which is regulating it? STAT6 and GATA3. STAT6 and GATA3, which are the regulating that part. Which are the major cytokine of that? Remember ASPA, IL-4, IL-5, IL-6, IL-9, IL-10, IL-30. And what does it do? Humoral response and eosinophil related diseases. Okay, that is more of the TH2 arm. Then you have TH17. I will come to other part. Okay, so TH17 is again something which is being regulated by a transcription factor called ROR gamma T, ROR gamma T. Okay, what are its cytokine? TH17, so IL-17 will come in picture, IL-22 will come in picture, and IL-26 will come in picture. It has two roles, one in autoimmunity, and second, it has role in protection against the bacterial and fungal pathogen. Fungal is quite important because we have agent against those IL-17, right? Secukinumab, ixekizumab and all. So with this agent, sometimes we see the increase in the fungal infection. So that immunity arm gets disturbed to that, right? Now we go to the other part, Th9 and Th13, and then we will come to T regulatory cell. So now Th9, again, your transcription factor is what? PU1, PU.1. And what is the cytokine? IL9. Th9, IL9, that is easy. Transcription factor is PU1. What does it do? It enhances inflammation in atopic diseases, right? Inflammatory bowel diseases and anti-tumor immunity also it plays role. So atopy, IBD, and the anti-tumor immunity, which is the main role of this Th9. Then comes Th13. So one of the cytokine is IL-13 with that, along with IL-4 and 5. What are the main transcription factor? GATA3. It's like Th2 ka some part. Okay. So that is a Th13 cell which is there. What does it do? It actually helps, or it really triggers the proper anaphylactic reaction. Okay. So allergen anaphylactic reaction, Th13 cell has a major role into that. Now see, this is all the pro-inflammatory pathways we are talking about. The inflammation increases. It requires a control. That is a T regulatory cell. What is the transcription factor of that? It's a FOXP3. It's a transcription factor of that. Can you remember one of the cytokine from this Treg, which is regulatory? We talked about one cytokine which does the regulation part more, IL-10. Quite important to know for that. Okay. And then comes the tolerance. Tolerance means, see, we are all talking about immune attack and immune system and all, but we have to tolerate few things and not to attack few things, not in just life, in immunity also. So you need certain mechanism which tells immune system, leave it. This is our body, leave it. This cell belongs to us, leave it. Okay, so first, leave it. Do not make antibody against that. That's central tolerance. Second, already things have gone little odd. Now just control it and don't do much. They are our part. That's called peripheral tolerance. It's a broad thing, but this is very easy way to just tell it from very starting. And the last part, which is my favorite, trained immunity. Trained immunity means there is a possibly misconception that the trained immunity can be uh, adaptive immunity because it gets trained properly with the spleen, lymph node and all. No, trained immunity in the real context is the innate immunity, which learns by epigenetic modification. Okay, so the epigenetic, transcriptional, functional reprogramming of innate immune system. Three things: epigenetic, transcriptal, and functional reprogramming, because we are getting trained of innate immune system. So we used to think innate is more robust, does not change much in life. No, it changes with the exposure part. So what you have been exposed to matters, right? Good textbook, good training. That is quite important when it comes to all this part. Okay. So that also happens. And that is something what we used to tell that uh, uh, the axe forgets, but the tree remembers, right? That kind of concept. That suppose we were eating junk food and living a very stressful life and all, and suddenly we tell Kinini, now I have to regulate. So 6-8 months goes a very regular life, and the body is like now getting better and all. But again, it bounced back. No, now I want to start again junk food. So the what damage initial junk food had done will be now enhanced if it is a restarted part, right? So are you telling you just keep eating junk food rather than doing this? No, no, no. It will again go very bad. Okay. But this is a concept to understand that the body, the innate system also has a memory. Okay. In a very broad concept, when we are discussing for the first time, we tell no, it does not have memory. But just remember, it has remembered the evolution completely. How can it be without memory? Right? It cannot be without that. So that's a concept of trained immunity. Forget too much concept and philosophy. Just remember, trained immunity concept applies to innate immunity. So I think I will close this here with the definition, and then the part two, I will start with the introduction very soon, and then it will go further. Thank you.